I personally feel kind of excited that the non-clear cell has moved on to the first day of this conference, and I hope that's reflecting all of the progress and optimism in the non-clear cell variants. And I want to mention that two of our speakers today, this is their very first kidney cancer meeting of any kind. So I think they came to the right place, and I know you guys will give them a really warm welcome and have lots of great questions. So with that, we will jump right in. Our first speaker is Jung Wook Park from Duke University, and the title of his talk is Defining Cellular and Genetic Factors for Renal Cell Carcinoma Subtypes. Thank you, Jung.
So thank you for giving me this opportunity to share our research here. Today I'm going to talk about this is a question like people address many times, like how the normal cell become cancer. We all know like oncogenic factors can make the cancer, but I'm going to address the same question, this old question, a little bit different way. So that's why I like to introduce it today. And next generation sequencing or the high throughput DNA sequence, the sequencing techniques like already identified so many different gene alteration in the kidney cancer. This is a little bit break but all the different subtype has a different the genetic alteration. So how are we going to address all of these genes like alteration? That's going to be the big question. So my labs, like we developed the primary human cell transformation system. So those systems already apply to the prostate cancer, bladder cancer, liver cancer, and what else? Like, so there are several like cancer we study. Now we try to apply it to the kidney cancer. So what you needed to understand the kidney cancer, we need a gene, but that's already like I show it here is like many genes are altered in the kidney cancer. We also need a specific cell type to transform the normal kidney cell. So I'm going to show you how we isolate the normal kidney cell in the next slide. So we have so many genes, like how are we gonna study all of these genes? Like if using the genetically engineered mouse model, it'll be difficult because you're gonna spend a lot of money to make the genetic model. So instead of that, we're using a lentivirus system, and then each gene will be barcoded. So that's the little piece of the DNA, so 8 to 10 base pair. So you can tell like which cell has which gene with the barcode system using the single cell DNA sequencing. And when we infect the kidney cell with the lentivirus, so then we culture them in organoid. So this time, cell gonna decide. It's like we can pull all the gene, or we can like subgroup the genes like you want to test for the specific kidney cancer subtype. So then during the organoid culture, cell gonna adapt it. It's like which gene combination they gonna like it or they don't like it. Also, this organoid culture system provide a nice environment for the primary cell. Because so when you infect the primary cell with the lentivirus, that's already damaged. So oftentimes people put it in the mouse directly, they're not gonna survive. So the organoid culture provide a nice environment for the primary cell. Then we put it in the mouse. And then because we're using the human cell, we cannot put it back to the human. So we're using a mouse model. And this concept, again, this is a very old concept, normal cell making cancer. But back to the old times, like people only use the one genes, like H-TERT or the SV40 large T, HPV oncogene like E6, E7, because it's hard to deliver the gene, like as many genes like they want. But we develop a systems like we can deliver it up to 10 to 20, 10 or the 20 different gene copy to the single cell. This technical advance, like we can study about like what the gene combination can make the cancer. So we have a gene, now we need a cell. It's like what cell we're gonna transform. And as you already know, kidney is not a single cell. Like kidney has an immune cell, T cell, B cell, monocyte, also has all different epithelial cell, the proximal tubule cell, collecting duct, and distal tubule cell. So this is the single cell RNA sequencing data. It's already published outside. So we look at the RNA-seq data set more carefully. Then we found this 4 cell surface protein. We can isolate the immune cell. Also, we can isolate the collecting duct, proximal tube, and distal tuber. So using this cell surface marker, we can isolate the specific cell type. As a proof of concept, so we get the fresh tumor, I mean, not the tumor, it's like normal tissue. Usually like people study the tumor cell, but we getting the normal cell or adjacent normal. And we chop it and enzymatic digestion, and then use the fax machine, and we isolate the collecting duct and then proximal tube. And then after we isolating, we put it in the 2D culture. We want to confirm like we actually isolate the 2 different cell type. And then we confirm these 2 different cell type, their growth rate is different. They also have different cell morphology. And we put it in the organoid with the lentivirus together, because we want to confirm whether this cell can accept the lentivirus. This time we use the 3 lentivirus, and then each lentivirus has color, green, red, or yellow. And also they contain the oncogene in the lentivirus. I'll show you in the next slide. So these 2 populations, like they grow well in the organoid. They start with a single cell, they making big chunk of the cell in the organoid culture, and then they show the color. When we put the 2 different organoids to the mice, only the 1 cell type can make the tumor, the proximal tubal cell. And then this organoid shows like express the AKT, MYC, and it's like we also inactivate the p53 using the dominant negative. And we also use RBSHRNA to suppress the RB1 expression. And the tumor looks like the clear cell carcinoma. And then we also look at the CA9 expression, because our target cell didn't express the CA9, but when they become tumor, they start express the CA9. So one of the beauty of our transformation systems, like now we know like a p53RB is not a common in the kidney cancer, but in this proof of concept experiments, like we put it in, but we can also take it out one by one and then see what happens. So that's the beauty of this system. So you can tell like what combination might be the driver, what combination might, it's like what gene may be the passenger. So the passenger may be determining the cancer phenotype, aggressiveness. So using this system, there are so many things we can address. First things like we can determine like what the cell is important for the tumorigenesis. I already showed the collecting duct didn't make the tumor with the same oncogenic vector compared to the proximal tube. So we can study like what's different in between the normal cell, what will be the suppressing the tumorigenicity. And from this thing, again, we can see what oncogenic combination can make the kidney cancer. That's also like bring us like what's the driver, what's the passenger. And the most exciting things like you can get from this assay is like we can capture the early stage of the tumor and the late stage of tumor. Then we can do multi-omics approach. Like you can do the transcriptional profile, epigenetic profile, or metabolic profile. So then we can understand what's like how the cancer is developed. At the end, like you will have a genetically defined disease model. So this one also can test for your gene of interest, also the drug. So like if you have a specific target for your drug, then you can make the specific disease model. So that's the things like we can learn from our disease model, I mean the transformation system. And I like to acknowledge all my lab members. Like my lab is still small, but they do work hard, like completes all the project. And I have a nice collaborations with Dan George and Andy Armstrong, like a nice medical oncologist. And then we also have the Ji-Yong Hong, like he provides us a small molecular inhibitor. So now we're making the model, also testing the drug together. So hopefully next year I can talk about some of the drug study. And I'm new in this field, so any comments, any question will be very helpful for my cancer study. Thank you.
Questions, make sure you're using a microphone because we've heard that people online are not able to hear. Yeah, I'll start with Ed, who has the microphone.
So weird. Oh, it's so weird. So this is fantastic. My question has to do with whether you can use the same system to sort of understand what kinds of environmental conditions might predispose you to transformation. So I'm actually imagining like medullary carcinoma, which I think there's some data that it arises in the context of really severe hypoxia. So for example, if you were to do this in the context of hypoxia, could you sort of tease apart how non-genetic factors promote or resist transformation?
Yeah, the hypoxia condition or like changing the microenvironments, like we can test it. So basically, it's like, well, yeah. So the one of the things, like again, it's like we isolating cell. So we can also isolating the stroma cell or the endothelial cell together and then culture it together, see if they can change the tumorigenicity. So you can also manipulate like those microenvironment can help the tumors like development. So this system's like basically you can do anything you want. So it's like bioengineering field. So this, we just need ideas like what things that you guys want to test it, then I can help it.
How was that? Do you have the?
My question is really about the genetics of the tumor that either your data or whatever, about the downstream biology combination of or whatever genetic I would like to model something that is really more a normal case there already been some but they just have not got to the point that it's language and I'm surprised that putting your pocket or something like DHL is still thinking that really massive CNI industry. So probably having some promiscuous firing of the CNI, and the cocktail is going to matter.
Yeah, so we do the downstream analysis. Like once we make the tumor, we just immediately submit to the RNA sequencing, and then we just compare how the tumor is similar to the human patient sample. Yeah. So surprisingly, it's like ours even has a 53 and RB, and it's still similar to the clear cell carcinoma in the gene expression profile. So, but it has a VHL. It's like we also try to suppress the VHL expression. Also, we try to introduce the VHL mutation together. So those things we can study. This is just a proof of concepts. Like actually those gene combination I applied to the many other cancer type too. And then we started from there. And then now it's you can test what of the downstreams things you want to test it. It's you can do it. So now we can make the cell line. So those things a very useful source for the kidney cancer.
There's a question from Bill back here.
I don't know. I'll build on Abhishek's comment. So first of all, I think the CA9 is probably due to AKT, which will upregulate HIF1. But I would really caution you. I mean, I would say what you showed us was sort of the bug, if you will, of this system, which is that combinations not found in nature can transform these cells. I think that's a bug or an undesirable feature of this system.
Right.
I think this is only useful when and if you can combine mutations that are found in real human kidney tumors and recapitulate the disease. So I'm not sure I heard the complete answer. I'm sure you're trying to inactivate VHL.
Yeah.
What happens when you inactivate VHL on these cells?
So VHL we tested. It's VHL mutation seems when we combine with these 4 gene combination together, it's they didn't initiate the tumor. So we still wondering, it's a VHL event, maybe the late stage of
No, it's categorically not a late stage event. We know that from patients who were born with germline VHL mutations.
Right,
we know from Charlie Swanton, it's an initiating event.
Right.
The issue, I'll tell you what the answer. The issue is almost certainly that it's a very rare cell within the kidney that is permissive for transformation by VHL loss. And you can probably configure your system to select for them. But unfortunately, when you do the experiment this way, as I understand it, what you identify are the most transformable cells with the most potent cocktails of oncogenes, whether they're found in nature or not. So I would just challenge you to think really hard about how we can get to for example, a clear cell model that has VHL loss and some of the other characteristic mutations associated with clear cell carcinoma.
So basically, it's like our model is like our approach maybe start from the like unknown combination that's possible, but we will narrow down like this combination actually is like happen in the human. So that's maybe we try to make any relevance like disease model. And then we try to like understand the BHL, like what cells maybe the BHL alteration can initiate the clear cell carcinoma. And in the single cell RNA-seq data, the proximal tubules, like there are like 4 different cell types, but we didn't like subtype them. We just included all of it now. So that's also the things like we need to further like separate the like cell type and then understand the like other PHL or other genetic alteration together. But it is a model, but the model also has some artificial fact, but we try to establish it and then we try to match it with the patient sample and then understanding from there. That's what we're trying to do.
Well, thank you very much. I think we're out of time for questions, but I hope that everyone will be following up with Jung Wook over the course of the next day or so. And
yeah, it's like
lots of
anything
feedback.
like please come to me like after this meeting. So I mean this section. So
Thanks.
Our next speaker is Kaushal Asrani from Johns Hopkins, who's going to be talking about targeting GPNMB in TSC and translocation RCC.
Good afternoon, and thanks for this opportunity. No conflicts. So GPNMB or glycoprotein non-metastatic melanoma protein B is a type 1 transmembrane protein. As you can see from its structure, it's got a pretty formidable structure. It has a large extracellular domain with multiple critical signaling motifs and a small intercellular tail that is important for kinase signaling. It's typically expressed at low levels in normal tissues, but it shows high expression in aggressive tumors like TNBC, gliomas, and malignant melanomas, where it's associated with a poor prognosis, where overexpressed. And most importantly, it has canonical M-box motifs in its promoter, making it a direct transcriptional target of the MIT TFE proteins, TFEB and 3. So there's extensive evidence for its role in tumor progression, both in a tumor intrinsic manner, as well as via modulation of the tumor microenvironment. Its extracellular domain is known to be cleaved in response to metalloproteinases, and in this way, it can modulate angiogenesis or immune function in the tumor microenvironment. It can also be secreted into the serum, where it can potentially serve as a soluble biomarker. It's normally located intracellularly, but in cancer, it shows increased cell surface expression, making it highly amenable to targeted forms of therapy. So previous groups have shown that GPNMB is expressed in various situations with constitutive mTOR activation in TSC, including CGAs, renal AML, and LAM, as well as in multiple MIT TFE-driven tumors. But the functional significance or its utility as a therapeutic target in TSC or TRCC are not known. So we and Dr. Henske's group have previously shown that in TSC loss, the transcription factors TFEB and 3 are constitutively hyperactivative and drive tumorigenesis. So we found that in HEC293 cells with TSC2 deletion, GPNMB protein expression is significantly increased, as so at the mRNA level, and genomic deletion of TFEB or 3 was sufficient to downregulate its expression in these cells. Expression was also increased by immunofluorescence in TSC2 and TSC12 null HEC293 cells. And in TSC heterozygous mice, which spontaneously lose a second copy of TSC and develop renal oncocytomas and cystadenomas with aging, with documental loss of TSC and increased S6, we were able to observe an increased expression of GPNMB in these tumors. We then examined its expression in a variety of human renal tumor subtypes. In normal tumors, it's expressed at very low levels, with the exception of histiocytes, which has been previously documented. It was also expressed at low levels in clear and papillary renal tumors, but highly overexpressed in a spectrum of translocation RCCs, including those Kathepsin-K negative cases. We then examined its expression in a spectrum of tumors with TSC1, 2, or mTOR alterations, such as ESC, EVT, and LOT, and conventional and epithelioid AMLs and picomas, where we found it to be highly overexpressed. We then examined its expression in cell lines derived from clear or papillary renal tumors with overexpression of the PRCC TFE3 transgenes. And its expression was significantly increased in whole cell and membrane fractions of the PRCC TFE3 cells. Membrane expression was also elevated by flow cytometry in panel C using a cell signaling antibody, and comparable results were obtained using CR011, which is a humanized monoclonal antibody against the extracellular domain developed by Creative Biolabs. Finally, we were able to see an increase in expression in PRCC TFE3 cells and xenografts by immunofluorescence and IHC compared to clear controls. Now, CR011 and its ADX counterpart CDX011 or glembutermab vedotin have been the subject of many preclinical studies. CDX011 has shown efficacy in preclinical models of melanoma and TNBC and had an acceptable safety profile, but subsequently did not meet its endpoints in phase 2 trials. Interestingly, a recent study has shown efficacy of CDX011 in preclinical models of TFE3 fusion RCC, suggesting that GPNMB could potentially be a viable candidate in DRCC. So for our first aim, we want to determine whether GPNMB can drive tumor progression or serve as a functional biomarker in the setting of TRCC or renal tumors with TSC loss. So to this end, we are generating cell lines with genomic depletion of GPNMB via CRISPR-Cas9 in the TSC2 null background and in the UOK 120 and 124 cell lines, and are currently in the process of obtaining and characterizing clones for further study. We also observe an increase in secreted GPNMB in the conditioned media of PRCC TFE3 cells compared to clear controls, and in urine samples of PRCC TFE3 KSP-3 transgenic mice. And we are in the process of further validating these results with additional assays. In our second aim, we want to determine if GPNMB can be targeted by alpha RP3, and if it can reduce renal tumor progression in TRCC or TSC. In the interest of time, I won't go into the advantages of alpha RPT, but it is a novel modality of treatment which has the capability to deliver high energy short range particles to tumor cells with a resultant increase in RBE. And that depends on finding a tumor associated cell surface target, making GPNMB an ideal candidate for this purpose. So we are currently in the process of optimizing the conjugation of CR011 with using either the DOTA or the DTPA chelator, followed by labeling of this antibody with indium-111 for imaging purposes, or the alpha emitter actinium-225 for therapeutic purposes. And although these results are preliminary, we have been able to isolate or obtain or conjugate and label the CR011 with indium DTPA with reasonably high yield and purity, and are currently testing it or evaluating its performance in immunoreactivity and binding assays. So in conclusion, GPNMB is overexpressed in TFE3 fusion RCC and in TSC deleted tumors in a TFEB and 3 dependent manner, but much more work needs to be done to determine its role in tumorigenesis or value as a biomarker, or or the efficacy of alpha RPT in this context. Finally, I would like to dedicate this work to the memory of my father, who passed away earlier this year from multiple myeloma. He's been my biggest source of inspiration and continues to be till this date. Finally, I'd like to thank my other family, my lab members, Tamara and everyone else in pathology, George and Anna from radiology, our funding mechanisms, and our collaborators at the NIH. Thank you.
Very interesting and very promising data, Dr. Asrani. We have been
Thanks.
following your work since you and Tamara published GPNMB. And we feel that there is potential value for this biomarker in surgical pathology-based tumor diagnostics as well. So we are trying to institute this immunohistochemical assay in our clinical lab at University of Michigan. My question is, since the MITF RCC now are 3 different subclasses, TFE3 rearranged or translocated RCC, TFEB translocated RCC, and TFEB amplified renal cell carcinoma, and especially the TFEB amplified renal cell carcinoma, which are now known to be poor prognostic actors. Would you anticipate differences or similarities in GPNMB expression in these 3 categories?
Based on the results of our recent publication, no, because we did see equal expression in all the 3 types based on both visual scoring as well as the digital quantification. So my answer to that would be, I guess, additional biomarkers are required to differentiate TFEB from 3 fusion RCC.
So that makes it even more promising, GPNMB.
Yes.
Thank you.
MIT TFE activated context. So just the 2 questions are, I noticed that you were looking into GPNMB as a driver of progression, but that seems like potentially a separate issue from whether it's a biomarker for TFE activated cancers. And how important do you think the answer to that question is, whether or not it's plays a role in tumor progression versus is it expressed on TFE rearranged or TFE amplified tumor cells? And then kind of the second question is, if it is activated, and clearly there's the EBOX motif in the promoter, and it's a very strong target of these genes, which are that whole family is expressed across a wide array of tissue types. What is known about the normal tissue expression of GPNMB, and what can that kind of tell us about anticipated toxicity?
Right. So for the first question, yes, we don't know enough about the biology, and those are potentially 2 different questions, which makes it all the more exciting, I guess, because it can serve as a biomarker, both soluble and tissue. But more recently, I think there's a very good review on this from Peter Siegel's lab, which talks about how it has roles as an immune modulator. So I suspect we're going to be addressing that question for many years, because even in the context of other tumors where it's already an established target, very little is known about how it actually drives tumor progression. So that's something we would have to work on, both in the tumor intrinsic manner as well as, with regard to the tumor microenvironment, which might be more important in cancer. And for the second question, what is that again? Sorry.
The normal tissue expression pattern.
Well, at least based on
you
our
can give a quick answer, because we actually have one more question.
Based on the staining, we have it's known that it's only expressed within. It has physiological roles in osteoclast differentiation and melanocyte maturation, and we don't see a very high expression profile in normal kidney. So to that extent, it's not much expressed. So it makes it a high value target.
So I just saw there are a couple of papers that suggest that this might be a HIF responsive gene, which would immediately explain why it would be upregulated during tumor progression as tumors become more hypoxic. So have you rigorously shown whether it is or is not simply a HIF target? And secondly, to the extent that you're pursuing a radiopharmaceutical approach, I assume you're looking very hard at whether any normal tissues express this at rest.
Right. So for the HIF question, we have not, but it's very interesting, and we will continue to look at that in all the models that we have. As far as what is the second question again? Sorry.
The radiopharmaceutical approach presumes that no normal tissues express this.
Right. So that's a limitation of our in vivo model right now, because the humanized antibody does not cross-react with mouse GPNMB. So we are potentially making an attempt to also develop a model where we can have the mouse antibody labeled, so we can then test it in our transgenic models. But in the absence of that, at least we will be able to address the question whether it's a viable target in the context of xenograft models.
Thank you very much. Thanks, everybody. Great question and answer. And again, this is Kaushal's first meeting, kidney cancer meeting, so I hope everyone will be following up with him. It's now my pleasure to introduce my colleague, Srinivas Viswanathan from the Dana-Farber Cancer Institute, talking about TFE3 fusions and translocation RCC.
Great, thank you, Lisa. So as this audience knows well, translocation RCC is an aggressive form of RCC, probably makes up a few percent of RCC overall in adults. And molecularly, it's driven by an in-frame translocation involving a transcription factor in the MIT TFE gene family, which is usually TFE3. And in their wild type state, these TFE genes modulate lysosomal biogenesis, autophagy, and the cellular stress response. But in the TFE rearranged cancers, these rearranged fusion proteins are constitutively active and constitutively nuclear, and that drives the disease. So importantly, the current therapies that we have for other subtypes of kidney cancer tend to be less effective in TRCC. And so this represents a major unmet need in kidney cancer research. So our lab is broadly interested in the biology and therapeutic targeting of TRCC, which falls into these three buckets. The first is comprehensive molecular characterization of TRCC, which involves the profiling of tumor tissues and models on various platforms. And that type of comprehensive molecular profiling has lagged behind in TRCC compared to other RCC subtypes. The second is a deep mechanistic understanding of the TFE fusion, which is the dominant and sometimes often the sole genetic driver and is universal across all TRCCs. And understanding exactly how the fusion works, I think, will be the key to identifying new windows for direct therapeutic targeting of that fusion. And then the third is to understand the pathways, the effector pathways that are either downstream of the fusion or synthetically lethal with it, to give opportunities for therapeutic targeting that can be tested in preclinical models and ultimately translated to patients. So for the sake of today, I'll focus on just a high-level overview of one project, which falls into the second bucket around the TFE3 fusion that's funded by the KCRP. So the TFE fusion is the universal genetic alteration that's found in all TRCCs, but there's actually quite some diversity in the fusion itself, both in terms of the fusion partners as well as in the breakpoints. So in terms of the C-terminus of the fusion, you can have the MIT TFE family member, and there are 4 family members: TFEB, TFEC, TFE3, MITF. And TFE3 is the most commonly rearranged. And then on the N-terminal side, there are a number of different fusion partners that can be paired with each TFE fusion gene. And in the case of TFE3, which is by far the most common, there are greater than 20 different fusion partners that have been described. And what's very interesting is that there's a very strong enrichment for RNA binding proteins amongst the TFE3 fusion partners. And we previously cataloged all the different fusion partners and breakpoints and TFE genes in our recent study. So to look at this more quantitatively, you can see in the bottom right there, we did an analysis where there's 25 different TFE3 fusion partners that have been described. So if you randomly select 25 genes from the genome and see how many of them are RNA binding proteins, and then do that 100,000 times, the red is the background distribution that you get. And you can see that if you do that experiment, you normally get like 0 to 2 RNA binding proteins. But what's observed is the blue line, which is 13 out of the 25 TFE3 fusion partners that have been reported are RNA binding proteins. So it's a really very, very significant enrichment for this class of TFE fusion partners. So that raises the question of whether both the fusion partner as well as the TFE side are functionally important for oncogenicity. So we looked at this initially with one of the common TFE3 fusions, which is no-no TFE3. TFE3 is the TFE gene in that fusion, and no-no is the RNA binding protein. And in a cell transformation assay, you can see that TFE3 alone in its wild type state does not transform the cells. That's the second picture. No-no alone doesn't transform the cells, but the fusion does. And so that suggests that you need both the TFE3 part as well as the fusion partner part. We then made inactivating mutations in both the TFE3 side as well as the no-no side. And as you see in the pictures and quantified in the bar graph, making inactivating mutations on either side, either fusion partner, reduces colony formation. So that suggests that there's really functional sequence-specific elements that are important both at the TFE3 side as well as in the no-no side. We looked at this systematically in the case of another fusion using CRISPR-Cas9 base editing. So Cas9 base editors allow you to, in a programmable fashion, make mutations at either specific cytosine or adenosine residues. And this can be a very powerful way to systematically mutagenize endogenous loci. So here we took a cell line which expresses the PRCC TFE3 fusion that was generated in Marston Linehan's lab and kindly provided to us. And we made a sgRNA library that tiled across this fusion so that we could mutate every cytosine residue that was potentially mutatable across that fusion. And so if those mutations are potentially deleterious, then the fusion will be less functional. And since the cell is dependent on the fusion, those cells will become depleted from the pool, and they'll be lower down on this graph here. So if you look at the graph on the right, you can see that making edits around the DNA binding domain of TFE3 leads to deleterious effects on fusion function, and those cells drop out from the pool. But interestingly, making edits on the PRCC side of the fusion, particularly over regions that are predicted to have a role in RNA binding, also is deleterious. So this suggests in a more unbiased fashion that there are important sequence elements on the TFE3 side as well as on the fusion partner side. And so we're currently in the process of trying to figure out what role those elements within the fusion partners may be playing. And one interesting observation is that there really are very different imaging appearances between wild-type TFE3 and the TFE3 fusions. So wild-type TFE3 in a fed state is mostly cytoplasmic, as you see at the left. The TFE3 fusions tend to be constitutively nuclear, but even within the nucleus are typically located within these punctate foci that are suggestive of their inclusion into condensates. When you make those inactivating mutations in the TFE3 fusion partner that disrupt RNA binding capacity, the fusion is still constitutively nuclear, but becomes more diffusely localized, suggesting that these condensates have been dissolved to some extent. And when you compare the interactomes of those mutant proteins compared to the wild-type no TFE3 fusion, there are many RNA binding protein interactions that are lost. So it's possible that there may be critical interactions that are brought in by the TFE3 fusion partner, perhaps with RNAs or RNA binding proteins that are important for conferring oncogenicity on the fusions. So to conclude, what I discussed is that there's actually substantial diversity in TFE fusions overall, both in terms of the breakpoint as well as the fusion partners. There's a very, very strong enrichment for RNA binding proteins amongst TFE3 fusion partners. And it seems that there are important sequence elements both within the TFE3 side as well as the fusion partner side. And it remains to be determined exactly what the mechanism is by which each of these sequence elements are conferring oncogenicity and whether that offers any opportunities for rational therapeutic targeting of the fusions. So I'd like to close by thanking members of the lab, especially the ones that contributed to this work, led by two postdoctoral fellows, Alessandro and Shata, as well as collaborators and funding sources. Thank you.
Thank you, Srini. Questions for Srini? Marston, can you wait for a microphone, please? Marston?
Thank you very much. Thank you very much. Very elegant work, very beautiful work. Let me ask you a question. We initially described TFE3 kidney cancer in '96, and we know that's a bad cancer. We know that it can spread when the tumors are small. We know that they can invade bad cancer. Now, in folliculin deficient, and you'll hear from Dr. Henske about TSC deficient, and maybe also about folliculin, but folliculin deficient, we hit the same pathway, TFE3, TFEB. Now, in patients with Birt-Hogby-Bay, we can see, and we recently with Andrea Balabio had a thing at IMBO, molecular medicine, on something we've seen over the years, and I kind of missed. I didn't really see it. I mean, it was there, but I kind of missed it. And there's the canonical tumors in BHD are oncocytic, chromo, basically, sometimes oncocytoma. But over the years, we've also seen some clear cell without VHL mutation that were really eosinophilic clear, and I didn't put two and two together, and some that look like TFE3, okay? Got it. Now, when we look at those, we have, 20 or 30 patients like that out of 700 or something. So it's not a big N, but they're aggressive cancers and they killed the patients. Now, when we look at those, we see TFE3 staining, we see TFEB staining, all right? Then when we look at the other tumors in Birt-Hogby-Bay, we see TFE3 staining, TFEB staining. In onco and hybrid, very sort of metastatic, can spread, but very non-metastatic cancers. I don't see I thought it'd be obvious to be a difference in TFE3. We're not seeing it. I mean, it's there, but it doesn't seem to be a bigger driver, if you know what I mean. Do you have any ideas what might explain that? I know. It's sort of. Where else can I ask this question?
I mean, yeah, I don't know exactly how to explain it, but the one interesting difference between all of the scenarios that you described and TFE rearranged cancers is the fusion partner. I think it's usually genetics is parsimonious. I think if it was just, if TFE rearranged cancer was really about just overexpressing TFE3 or TFEB, we would see focal TFE3 amplifications, or we'd see N-terminal truncations that remove the NLS and make it constitutively nuclear without having the fusion partner, but that's not really observed. So I think that the phenotype that you see in TFE rearranged cancers may actually have function coming from the fusion partner, as well as the TFE3 side, which may be driving a different phenotype than, for example, folliculum loss or BHD, where you're just seeing activation of the wild type. That's one thing. And then the other thing is kind of the interplay between the other MIT TFE family members and whether there's co-expression that might be driving the phenotypic differences as well, I think.
That's a good insight. And that may give us some ideas. Thanks.
Yes, Abhishek.
Srini, a couple of questions in terms of the localization patterns that you're seeing. Almost looks very similar to what paraspeckles look like, which are enriched in RNA processing proteins at sites of transcription. Is there any evidence to indicate whether the complex can recruit nascently synthesized RNA versus RNA that is just not being actively expressed, but still not being translated, but has been expressed and is sort of stored in the cell. In line with that, let me finish a thought. The combination partners that you have, do they all show this speckled appearance in cells?
Yeah, so those are two great questions. I think for the first one, in terms of the appearance similar to paraspeckles, that's very astute. And I didn't get into this for the sake of time, but NONO is a core paraspeckle component member. There are 3 core members, NONO, SFPQ, and PSPC1. SFPQ is also a recurrent TRCC fusion partner. And if you do co-localization studies, the fusion, the NONO TFE3 fusion is co-localized with the other endogenous paraspeckle components, suggesting that it is being localized there. The other fusion partners, most of them also show various types of speckled appearances, although sometimes a little bit different than the NONO one. But typically not diffuse nuclear expression. So most of them tend to show some kind of, that we've checked, some kind of speckled appearance.
We have one more question over here.
Canonical fusions for the MITF RCC, like you showed out with the PRCC and NONO as well. Are you seeing similar RNA binding capacity with ASPL also, which is likely the commonest fusion for TFE3 RCC, ASPL?
In terms of the imaging pattern?
Yeah.
Yeah, so we haven't looked at the ASPS TFE3 fusion in as much detail, but it does appear to have somewhat distinct appearance from the clearly annotated RNA binding proteins, but still potentially speckled.
And thank you. And if I'm understanding correctly, and I think I'm curious what you feel, and I think Berthold was also mentioned, I feel that follicular deficiency, MITF overexpression, and mTOR TSC pathways, they have such a complex interplay that we won't be able to really find biomarkers or unique downstream functional effects or singular biologic or therapeutic effectors, because the interplay is so complex. For example, GPNMB, like shown by Dr. Asrani, I think is overexpressed in both MITF RCC as well as the mTOR TSC RCC. And I think that's another example. And I feel all of these are just caught in a positive loop feedback phenomenon. Thanks.
Yeah, I agree with that. And also getting back to Dr. Linehan's point, I think another difference is you lose the N-terminus in the case of the fusion of TFE3, which has a lot of the phosphorylation sites that are regulated by the mTOR pathway. And that may be the difference between TFE rearranged cancers and other cancers in which TFE, the wild type form of the TFE genes, are upregulated.
Maybe one more quick question from Lawrence. A question and a quick answer. Can we have the microphone again, Susan, up front? Can you repeat the question? So people can hear online.
TFE3 amplification. Yeah, so I think TFEB amplification and TFE3 amplification is interesting and potentially, I mean, one would expect for it to be molecularly different, but I think we need to probably treat all of these as separate entities because biologically we predict it to have different effects than these in-frame fusions. But I think it remains to be seen.
Thank you, Srini.
And our last speaker in this session is Lisa Henske.
Thank you. This has been a really fun session. So I'm going to be talking about TFEB and TFE3. So that's a good segue. I want to start by acknowledging the people who I work with, especially Nico Alessi and Damir Habibulin, whose unpublished data I'll be sharing today, and Michelle El-Shaweri, Samer Salem, and Melissa Dow, who are all here today, and the amazing team I get to work with, including David Kwiatkowski and Carmen Priolo at the Brigham and Women's, and the team at the Dana-Farber, which includes Srini and especially Tony, who is just incredibly generous and visionary in making good things happen every single day. So I'm going to tell you about three things today. First, about a little bit about tuberous sclerosis associated renal cell carcinoma. Secondly, about how TFEB is very important in the pathogenesis of renal tumors in TSC, and we think also the therapy. And then lastly, some links to Birt-Hogg-Debay syndrome and chromophobe kidney cancer. So tuberous sclerosis affects almost every organ in the body, but my lab is especially interested in the lung manifestation, LAM, and in the kidney tumors. In the kidney, the most common tumor is, of course, the angiomyolipoma, which are benign fat-containing tumors. You can see the fat in this tumor right here. But there also are renal cell carcinomas, which are often chromophobe-ish. They sometimes are called hybrid oncocytic chromophobe tumors. Chromophobe tumors, as you know, have this plant-like plasma membrane, a sort of wrinkled raisin-looking nucleus, and in tuberous sclerosis, these tumors occur at a young age. Average age is 36, and they have a female predominance of 2:1, and they've even been described in children. So in TSC, the conventional understanding is that the TSC1 and TSC2 proteins function as a heterodimer to inhibit the small GTPase Reb, which is required for the activation of mTOR complex 1, so that in tuberous sclerosis deficient tumors, there's hyperactivation of Reb and activation of mTOR. And of course, mTOR can be inhibited with sirolimus or everolimus. And the way we usually treat tumors in tuberous sclerosis is using an mTOR inhibitor. Both Afinitor and rapamycin are approved for different indications. And you can see here what happens. The tumors shrink by about 50% in 1 year of therapy. And then that red arrow shows you when the therapy is stopped, the tumors grow right back. And we don't really understand all the reasons for this, but the result is that if you have tuberous sclerosis and you need therapy, you'll be on an mTOR inhibitor for a long time, maybe forever. And there now are children who have been on mTOR inhibitors since early childhood, since we first discovered this. So the project I'll tell you about today started with the observation by electron microscopy that lysosomes are about threefold enriched in tumor cells from mouse models of tuberous sclerosis. We also, as was already mentioned by Dr. Asrani, we see increased lysosomal genes and proteins in tuberous sclerosis. I'm just showing you one example here. You're seeing TSC2 deficient mouse embryonic fibroblasts, and you can see upregulation of the Niemann-Pick type C protein. That's a classic lysosomal protein, and cathepsin K. We also see this by immunohistochemistry in the tumors, and I'll just show you this one example because this is a sporadic tumor with a TSC2 mutation, and it's one of these newly defined subtypes from the WHO of a eosinophilic and cystic renal cell carcinoma. And this is staining again for NPC1, and you can see the tumor is markedly positive. And we see this very strongly also in all tuberous sclerosis associated tumor types that we've looked at. So this led us to be thinking about TFEB and TFE3, and we're very lucky to have the input and collaboration with Srinivas Viswanathan. So you've already heard a bit about TFEB and TFE3, which are considered master regulators of lysosomal biogenesis. And they are regulated by many different pathways, including mTOR complex 1, which phosphorylates TFEB at the site that's circled there in blue leading to its cytoplasmic sequestration. What's very surprising and unexpected in tuberous sclerosis, we've seen this, it's also been seen by Dr. Asrani and by Jim Brugarolis, is that despite high mTOR activity in TSC2 or TSC1 deficient cells, TFEB and TFE3 are in the nucleus. So you see this here in HeLa cells with expressing GFP tagged TFEB with downregulation of either TSC1 or TSC2. It's really obvious that TFEB is in the nucleus. We see the same here with TFE3. And using a reporter assay that was developed by Srinivas Viswanathan, we can see that GPNMB reporter activity, which was the protein that Dr. Asrani was speaking about just a few minutes ago, reporter activity is markedly upregulated in TSC2 deficient cells shown in red, and also in folliculin deficient cells, the Birt-Hogg-Debye protein shown in green. So the question is, does this matter? TFEB is definitely in the nucleus. Does it have any effect on tumorigenesis? Well, the answer is it definitely does. So this is unpublished data from Nico Alessi and Damir Habibulin. And what you see here first are just a bunch of control kidneys from a kidney-specific CRE model, including TFEB knockout alone. If you knock out TSC2, you see enlarged cystic kidneys. And if you knock out both TSC2 and TFEB, the mice are normal. They happily live a long, healthy life. They can reproduce. Everything is fine. So, and this shows you just histologically what we're looking at. This is day 90. The tumors are markedly cystic if you knock out TSC2, and knocking out TFEB just fixes it completely. So it looks like TFEB drives renal disease in mouse models of tuberous sclerosis. We see the same in another model. It's quite unexpected, I would say, because we would have not even guessed that TFEB would be in the nucleus in this setting. And the same thing happens in Birt-Hogg-Debye syndrome. So we actually have one of the Kidney Cancer Research Program partnership grants with Andrea Bellabio to address this in both tuberous sclerosis and Birt-Hogg-Debye. And this is his work published in Nature in 2020, showing that if you knock out the Birt-Hogg-Debye gene, folliculin, the tumors are markedly, kidneys are markedly enlarged. And if you knock out both folliculin and TFEB, they're normalized. So in the last few seconds here, I want to tell you a little bit about chromophobe kidney cancer and how this might connect. This is showing you a large chromophobe renal cell carcinoma. I'm sure you know that chromophobe is really quite different from clear cell in virtually every way you can think of. They have a high rate of mitochondrial mutations, loss of one copy of multiple chromosomes, few driver mutations. They arise from these really cool cells in the kidney called intercalated cells, and they have markedly elevated glutathione and sensitivity to ferroptosis induction. So it turns out that NPC1, this Niemann-Pick type 1 protein that I told you about a moment ago, across the whole TCGA, the highest levels are in chromophobe kidney cancer, shown here with the blue arrow. We also know from really beautiful proteomic work done by David Meyerhofer's lab in Germany that the lysosome pathway is strikingly upregulated in chromophobe, including GPNMB, which had the third highest fold change. So we wonder whether TFEB and TFE3 might be the missing link in several different forms of non-clear cell kidney cancer, including tuberous sclerosis and Birt-Hogg-Debye associated, maybe in chromophobe, and oncocytomas, definitely in the TFEB and TFE3 rearranged, of course, and also in these rare subtypes that are caused by TSC2 mutations, such as the eosinophilic and cystic tumor. We don't know whether some of these or all of them arise from these interesting intercalated cells, but it seems possible that TFEB and TFE3 are playing a role in all of them. So my take-home messages are that kidney cancer and tuberous sclerosis and Birt-Hogg-Debye often has this interesting oncocytic chromophobe histology and upregulation of the lysosomal targets of TFEB and TFE3, which seems to be a theme through this session, that in mouse models of both tuberous sclerosis and Birt-Hogg-Debye and work done by us and by our colleague Andrea Bellabio, TFEB knockout fixes completely the kidney disease, and that lastly, TFEB and TFE3 targets, including lysosomal genes and proteins, are upregulated in sporadic chromophobe kidney cancer. Thank you very much.
Oh.
These are the great talk.
I think it'll automatically turn.
So you did show that the TFEB knockout reverses the phenotype of the KSP-CRE TSC2 mice, which are predominantly cystic, does it also reverse like more solid lesions seen in the T2AJ mice? Because and the second question is there no role in this work and by previous work by Dr. Balabio, TFEB alone is enough to fix the abnormalities. Is there no role for TFE3 in this context?
Yeah, so the first question about solid tumors in TSC, we don't know. We have not checked any models in which solid tumors are the primary phenotype. In terms of TFE3, it's really surprising that TFEB is able to completely fix the phenotype, but we haven't looked at TFE3 yet, so I don't know the answer to that.
Tony? Actually, I think we have a question over there first.
You pointed out that it's kind of contra to what we would think that the TFEB would be in the nucleus. It's, with increased mTOR activity, you would expect more of it to be in the cytoplasm. So I'm just curious, if you give an mTOR inhibitor to those mice, what happens to the TFEB in terms of its translocation?
Yeah, it's very perplexing. So the mTOR inhibitor taurin keeps TFEB and TFE3 in the nucleus, but the mTOR inhibitor rapamycin actually moves it out of the nucleus to the cytoplasm. It's paradoxical and very mysterious, and we don't really understand the mechanisms for that, even though we would like to understand them. It's a great question.
Talk, your focus on chromophobe RC commendable. So especially with novel combination with immunotherapy, we really have excluded chromophobe. We have not seen responses with immune checkpoint inhibitor at all, except perhaps when patients have extensive sarcomatoid dedifferentiation. But the work with Laurence and Merck and Jolie showed finally with pembrolizumab 28% response rate. So my question for you is, it's a cold tumor. We're working on something together, but what other immunological pathway? We've seen some elegant work from Memorial with IL-15. How are you going to go through that angle?
Yeah, we're very excited about the work that you just mentioned from Ari Hakimi and others, showing that ILC cells, which are innate lymphoid cells, kind of like natural killer cells, are increased in the chromophobe tumors and seem to be responsive to IL-15. So it might provide an angle for targeted therapy that would be really exciting, and we hope that will move forward.
Lisa, I always had a curiosity, and just as a TSC expert, whenever we sequence AMLs, sporadic mostly, we've only seen TSC2 mutations. Is there any reason why that over TSC1, or any insights into the complex itself is why that might drive tumorigenesis? Just curious.
Yeah, so we see the same in angiomyolipomas, you're right, it's always TSC2, and the same in the sporadic form of LAM, lymphangioliomyomatosis, it's always TSC2. And one very naive explanation might be that TSC2, of course, contains the GTPase activating domain for Rheb, where TSC1 is more of a scaffolding protein. So it might be that in the absence of TSC1, there's just enough TSC2 that's able to do its job that makes it less severe. So overall, the phenotypes in humans with germline mutations are exactly the same, but the severity is consistently more in TSC2 mutations.
Dr. Rathmel?
But in your talk, as well as with some of the others, is about the young age of patients with these particular genetic abnormalities. And if you have any thoughts about, is it just simpler tumors, and that's why they can arise at teenage years, 20s and 30s, or if you think there's something more sort of developmentally switch regulated?
It's a great question. I don't have any answer. I don't know if any of my co-speakers have an answer for that.
We did 2 papers with Al Knudsen before he passed away, and we showed, and one with Dr. Henske, and showed a haplo effect in both VHL and TSC in the normal of TSC patients who had surgery for AMLs or tumors, and same thing with VHL. So could that play a role in the earlier onset, or the fact that every cell in the body, every cell in the kidney will say, has one hit, and it's a lot easier to lose the other and get them earlier? I don't know, but I don't know if you have any other thoughts about that.
Yeah, I think that could be.
I was going to just add one thing about, you mentioned something about TF3 versus TFEB. And again, in worked with Andrea Blabio, we've shown in 10 that folliculin regulates TFE3 translocation and phosphorylation and turns up GPNMB, and you can see that in the human tumors and all that. But and that inhibited growth in vitro. But when TFE3 is knocked out in the KSB3 follicular knockout mouse, it doesn't really have a big effect, whereas TFEB has a huge effect. But in the human cell lines, when you knock it, and we just think that that's an artifact of KSB3 sort of expression, we think. In the human tumors, when you knock out TFE3, it totally abrogates their tumorigenesis, just like it does TFEB. So our sense is that they're both kind of equal. But when you knock them both out in the animal model, you get a better effect than either one alone for sure. So the sense there is maybe they're additive, but.
Yes, that's an important point. Thank you.
One more.
One last question. Dr. Kinnear.
He's here. Microphone.
So, it's good. It's a great talk, and Srini. So you published last year about ferroptosis and chromophobe, and it seems like it's also a pathway that we saw also in RMC. We published recently on this in Nature Com. And so do you think this is a conversion pathway for, TFE3, TFEB, many of these variant histologies, non-clear cell?
That's a great question. It doesn't, I guess, based on Srini's work, it might not be actually. It might be going in different directions. Ferroptosis may be important, but in the setting of chromophobe, we think that inducing ferroptosis is going to cause tumor cell death. And I'm not sure that's the same for the translocation. So I guess that's an area that I think needs more investigation. Just on a very quick note, if anyone is trying to grow chromophobe cells in culture, we just had this kind of brainstorm last week that maybe if you were to add exogenous NAC or glutamax, which is a cell permeable form of glutamine, maybe you would prevent the cells from undergoing a crisis in cell culture or organoid culture related to their need for high cysteine uptake. So if you have any patients who you might want to be culturing, maybe let me know and I'll try to help think that through ahead of time. Thanks.
Great. So I think we're thank you everyone for a great session. I'll just echo Lisa's comment at the beginning that it's great to see all of this effort and enthusiasm devoted to non-clear cell. Thank you.